Welcome back to our next biology class. This is continuation of the chapter Human Health and Disease. And today we'll take up the topic immunity. Students, in our everyday life, we come across a wide variety of infectious agents. Yet, we do not develop the disease all the time. It is very rarely that a person suffers from any critical or that sort of illness, whereas we are exposed to these infectious agents every day. Have you ever thought about why is it so? It is because of the complex system that exists in our body in the form of immune system. The term immunity is very familiar to you all because nowadays in every advertisement you will find the term immunity boosters, which claim to increase your immunity so that you don't suffer from COVID-19 or any other infection. Do you know what this immunity is? Let us see in details in this particular module. The term immunity means natural or acquired resistance to a particular pathogen and its infection even after exposure to a good dose or infective dose of the same infective agent. And this is brought about by a complex system in our body known as the immune system, which has both cellular and chemical components. To understand this system, let us first see what are the different types of immunity. There are two broad categories of immunity: innate immunity and acquired immunity. Innate immunity is also known as inborn immunity, genetic immunity, familial immunity, etc. It is a non-specific immunity and comprises the major and the primary portion of our immune system. Now, this inborn or innate immunity is contributed by various factors, various cells, and we will see them in details. The first line of defense of our body is formed by the physical and physiological barrier. The cellular cytokine complement system, they form the second line of defense. And the third line of defense is formed by the acquired immunity. What do we mean by physical barrier? These are the anatomical barriers which prevents the entry of the microorganism in our body. Like the first barrier is the skin. You all know skin does not allow anything to enter through it if it is intact. Microorganisms can enter only when the skin is ruptured or disrupted. The second anatomical barrier is your nasal mucosa. The nasal mucosa, with the help of the cilia and the mucus, they drive or they trap any of the pathogen that comes in its contact. Then you have the different lining of your digestive and the respiratory system that forms the anatomical or physical barrier. The second one are the physiological barriers, which comprises of the friendly microbes that harbor our body and that drives the harmful pathogens away. The body temperature is set so that it does not allow most of the organisms to grow in that temperature. And also that when a pathogen enters your body, the body sets its temperature to somewhat elevated temperature in the form of fever, so that the pathogen which has entered your body cannot survive in that particular elevated temperature. As because all organisms have an optimum temperature at which they thrive the best. Above and below the optimum temperature, the activity of the organism decreases. The acidity present in our stomach can even melt a metallic blade. So think of the organisms that come in contact with this acidity. They get destroyed. Similarly, our ear produces earwax or cerumen, which actually protects the internal environment of the ear from entry of the pathogen. Similarly, the sweat and the sebum that are produced by our skin glands are our physiological barrier which stops the entry of the microorganisms or kills the microorganisms which even comes in contact with it. The third but the most important one are the cellular barriers. Cellular barriers are formed by the macrophage or phagocytotic cell, to be more precise, phagocytotic cells and the NK cells. Phagocytotic cells are of two types, macrophage cells and the neutrophil. Many times you have heard that WBC are the cells which fight the infection. So which cells or which type of WBC fights the infection? The primary role is taken by the lymphocytes and the neutrophil. The neutrophils or polymorphonuclear lymphocyte, as they are called technically, are the major phagocytotic cells of the blood. And macrophages are the cells of reticuloendothelial origin. What do? They go to the site of infection, engulf the microorganism and kill it within itself by pouring off the lysosome that is present in the cell organelles. The NK cells or natural killer cells are the small lymphocytes without B or T markers. They increase the permeability of the cell to water by forming aquaporins or pores in their cell membrane so that the water can enter into the cell. As a result, the cell swells up and it gets disrupted. So natural killer cells are also a very important component of the innate immunity. Next are the cytokines. Cytokines are the non-cellular, non-antibody chemicals or protein molecules of a body that helps to kill the infective agents. There are other barriers also, which we call inflammatory barrier. Whenever there is infection, triple response occurs at that particular site, the site of infection, in the form of redness, swelling and pain. This triggers a complement system or a complex system of chemicals in the form of, say, histamines, prostaglandins, interferons, which actually destroy the infective agents. Complement system is a system or a cascade system of, say, 30 odd proteins which work in a cascade manner and destroys the infective organisms. As I said, they all are non-specific. They have the same response to any of the infective agents. Interferons are especially important to fight off the viruses because, virus infected cells, they release this substance known as interferons, and it makes the surrounding cells non-infective to the viruses. So it is against the virus, body develops its own immunity in the form of interferons. But the specific immunity against each type of microorganism separately is provided by the acquired immunity component of the immune system. As I said, innate immunity or inborn immunity is the type of immunity with which we are born with. It is present right from the time of the birth of an individual. In contrast to it, the acquired immunity is the type of immunity that we acquire or that we develop in due course of our lifetime. This is also called adaptive immunity, and this is the specific type of immunity. It adds up to the innate immunity. Now, this type of immunity is only present in vertebrates and takes time to develop. It needs an exposure, and after that several days. The first encounter with the pathogen produces a mild response. This is known as the primary response and takes several days to develop. But the second encounter with the same pathogen produces a more vigorous and a fast response. And this is the basis of the vaccination. Now, the major unique characteristics of the acquired immunity is number one, specificity. That means it is very specific. It can identify one pathogen from the other. So, it depends on the pathogen. Diversity. There are diverse types of response to against each and every pathogen. The third, it has the ability to distinguish between the self cells and the non-self cells. And that is the basis of this immunity. It can identify which is my own cells and which is a pathogen. And the fourth one is memory, which is the most important attribute of this type of memory. As I said, the primary response is a weak response that produces memory cells, which makes the second encounter with the pathogen easily identifiable, and a more vigorous, faster response is generated in case of the second infection. The acquired immunity is brought about by two major components of our immune system. Number one, the cell-mediated immunity, where the cytotoxic cells or the T cells of our immune system, they destroy the pathogen which has already entered in the host cell. And it also works against the transplanted organs. Most of the time, we need to match the organs of the donor and the recipient when a transplantation of organs takes place. This is because the cell-mediated immunity gets activated in case of the transplantation, and if the match is not proper, it triggers a vigorous immune response and rejection of the grafts. The second component of the acquired immune system is the antibody mediated immunity, or more commonly called humoral immunity, which is present in humors or the secretions of the body. Most importantly, the lymph, the plasma fluid, and the blood. This is brought about by the antibodies. Antibodies are the immunoglobulins, a type of proteins that are produced by special lymphocytes known as the plasma cells. These plasma cells secrete antibodies. Antibodies are of 5 types, namely A, D, E, G and M. Antibody A is most common in the mother's milk and in colostrum, the first milk secreted by the mother, which actually provides immunity to the baby, and that is why it's so important for the newborn baby. IgD is mainly important for the allergic response, identification of toxins and the chemical allergens. IgA is responsible entirely for the allergic responses. IgG is the major antibody and constitutes about 75% of the total antibody population in the body. They can cross the transplacental barrier. And IgM are the first antibody to reach the place of infection. They are pentameric molecules, means 5 antibody molecules are attached to each other to form a pentamer. Now let us see what is the structure of this antibody and how it brings about the immunity in our body. Now, let us see the structure of an antibody molecule. An antibody molecule consists of 4 polypeptide chains. 2 heavy chains, which are represented as the longer chains, and 2 short, light chains. The heavy chains are joined to each other with the disulfide bonds, as well as the light chains are joined to the heavy chains with the disulfide bond. Not only this, it is the disulfide bond which brings about the bending of the heavy chains. Now, as I said, antibody molecules are specific for specific antigen or specific pathogen. And for this, an antibody molecule has a constant region which is same for all antibody molecule and terminal variable region which is specific for each and every antigen. So, the variable region varies for each and every antigen, and this is what gives specificity to the antibodies. Now, the antigen binds to the antigenic binding site or antigen binding site of the terminal portion of the heavy and light chain. Heavy and light chain, the antigen binding site, which is a part of the variable region. Now, an antigen has an antigenic determinant on the surface of the antigen. It is through the antigenic determinant that the antigen binds to the antigenic binding site of the antibody. Once the antigen is bound to the antigen binding site of the antibody, it brings about 4 types of responses. The first response is the neutralization. Once the antigen is bound to the antibody, it can no longer move or infect any cell. So its activity is neutralized. Second, it brings about agglutination or clumping of the antigen molecule. Once the antigen is bound to the antibody, all the antigens, they come together and clump and become heavy. The third is adherence. They adhere to the antibody and all they come together making it a heavy molecule, followed by precipitation. That means the antigen-antibody complex becomes heavy and settles down. After settling, the lysis of the cell occurs, one with the help of the lysine antibody or with the help of the cytotoxic T cells, which bring about the destruction of the antigen-antibody complex. And this is how the antibody provides immunity against the pathogen. All the pathogens have different antigenic determinants and hence they trigger the formation of different antibody molecule. Thus, this is a specific response generated against the pathogen.